So focusing more on taking mutation data and trying to analyze that mutation data. Maybe I'll give people a sec to come in. Okay, all right, well, I guess we'll just roll. All right, so I work in the Hammerbacher lab at Mount Sinai Medical School in New York. And so our PI is Jeff Hammerbacher. He's a gentleman in the upper left over there. I guess I'm going to give him a sec to get everyone in. Awesome, thank you. Okay, so some of you might know of Jeff. He's a co-founder of Cloudera and worked at Facebook. So he has a very computer science background, sort of a peculiar thing that he's working in a biomedical research lab. And so a distinguishing characteristic of our lab is that our biology expertise is pretty thin. I mean, we've been learning while working there, but very few of us come from bio backgrounds. We're primarily computer scientists, programmers, data scientists, because that's become a term now. And we're taking that domain expertise and trying to find good collaborations that need those kinds of skills. And so we have kind of a split focus of building software, building open source software, putting everything on GitHub, nice unit tests, sort of the higher standards of quality than you often find in bioinformatics projects is what we're aiming for. And some people have used bioinformatics tools and they know what I'm talking about. And so the motivation for a lot of the software are research projects. So we're not really a software development lab. We write a lot of software, but the motivation for that software is often sort of guided by a desire to have some sort of clinical impact, because we're in a medical school, which is associated with a hospital, and we have an opportunity, when we're lucky and everything goes well and you put in a lot of hard work, to actually affect clinical outcome. And so that's sort of the gold standard. It's hard to get there, but once in a while you get this opportunity to change the way that patients get treated. That's what we're aiming for. And so example of some of our projects are, these are really the genomics focused ones. So we're looking at trying to predict response to immunotherapy. You can sequence a cancer and then try to determine whether or not the, after a cancer has been excised from a patient, will the residual cancer that might turn into metastasis respond to an immunotherapy? And you try to do that from primarily either the expression or mutation data. And so another project we're working on is looking at samples from ovarian cancers and try to determine if chemotherapies, particularly platinum chemotherapies, what their effect is on the spectrum of mutations in those cancers. So we're looking at sequential biopsies or sequential resections and the sequence data that comes from those. And what's maybe our sort of most interesting current project, this last one, is a collaboration with Nina Bardawaj's lab at Mount Sinai. We're working on a personalized cancer vaccine where, based on the mutational content of a patient's cancer, we have an automated pipeline written mostly in Python, which predicts what possible vaccine contents might be therapeutically beneficial. So trying to reprogram the immune system to recognize mutated proteins and attack cells presenting those proteins. So all of these are, highlighted the word mutation everywhere because that's sort of the centerpiece of all these projects. We need to be able to work with the mutations from a patient's cancer. So a very quick overview of how gene sequencing works for cancer. For those that aren't familiar, I'm going to do a terrible job of this. So the basic idea is you're trying to compare the differences between normal cells and cancerous cells. So you get a sample of both. A piece of the tumor and often a blood draw is representative of what's normal. And then you extract the DNA from those samples and you fragment the DNA into small pieces, a few hundred nucleotides long. And then you sequence bits of those pieces so you get these reads, maybe it's essentially a giant text file with lines that are about like 150 characters long, full of ACTGs. And then through a bunch of bioinformatic tricks, you get a summary of what were the differences between the normal and the tumor DNA. So here's an example of what one of those differences shows up like. So at the top here, we have the reference genome sequence. So a lot of this work is done relative to a reference genome. This isn't really any one particular person's genome. It was collected from, I think, 20 volunteers in Buffalo, New York, and then sort of assembled into just, here's our reference point in genomic space, and then we describe mutations relative to that reference point. So we have a little bit of the sequence of the reference at the top, and then there are reads from the normal DNA, and they're grayed out where they agree with the reference. And then there's one spot where the tumor DNA, many of the reads disagree with the reference. And so that's a somatic mutation, a cancer mutation. And then the summary of this kind of information ends up in a couple of file formats. There's two that we use primarily. One is VCF, variant call file, I think. And all the payload is at the bottom left over there. You could see the chromosome position, the reference nucleotide, and the alternative nucleotide that was actually found. And so that's like a tremendous condensation of all the data generated by sequencing. You go from hundreds of gigabytes of these raw reads down to some several hundred lines in a VCF file. And so another file format we work with is called MAF. And so this mostly comes out of the TCGA, the Cancer Genome Atlas. And so this MAF has, it's a little more tailored for cancer genomics. VCF is very general. You can use it for pretty much anything where you're looking at differences in genetic content, whereas MAF is like hard-baked for a notion of there being a normal and a tumor allele. Okay, so that's sort of the background, and now getting to the actual tools that we've built. One of them is PyEnsembl, and really has nothing to do with the mutations themselves, but it's the baseline of information about the reference that you need to then figure out anything about the mutations. So there are a couple databases of metadata on top of the genome. They tell you where the genes are, where the exons are, what their sequences are, and things like that. And so one of them is Ensembl. They have a very clean sort of automated pipeline for assembling all the metadata. And PyEnsembl is a Python wrapper for all their various file formats that they generate. So you can use PyEnsembl to download ensemble data, and then it has Python objects that represent genes and transcripts and exons and things like that. And then Varcode is a library used to actually interact with variant information, with mutations. And you can use it to just compare sets of mutations, or you can use it to actually predict the impact of those mutations on proteins. Sort of the most, the notion of an impact of a mutation can be really broad. It can do lots of things. So we're looking at the smallest notion of what's the effect of a mutation, just in terms of does it affect the protein sequence, and if so, what is the new protein sequence? All right, so to get you started, you first have to install PyEnsembl and Varcode, and then download Ensembl reference data, which takes a while. It's a few gigabytes, and it gets built into some SQLite databases and index. So you spend about 10 minutes waiting for that second command to complete. And you have to choose an Ensembl release that corresponds to which human reference your variants are aligned against. So there's two really common ones. So at the bottom you can see if it's one genome, you use Ensembl 75. If it's a different, you use Ensembl 80. And then here's an example of actually using PyEnsembl to get some information. So we're talking about cancer genomics, very well-known cancer-related gene is TP53. This is the guardian of the genome. It has a lot of functions related to preserving cell fate and keeping cells kind of normal, and then detecting if they're damaged and initiating apoptosis if they're messed up. And so we can look up TP53 by name. And so there are different Ensembl releases at the top level namespace of PyEnsembl. So we grab for one of for a release associated with a particular genome, we call genes by name TP53. Gives us back a list of gene objects, because sometimes a gene comes in many copies. And then there's only one TP53, so we grab that out of the list, and we can get different properties of that gene. So you can find out that it's a protein coding gene, you can get a list of all of its transcripts, and you can spend a while just exploring the properties on all those objects. And also, importantly, you can get the sequences for different transcripts of TP53, which becomes useful for Varcode. So you can get the nucleotide sequence of the DNA, you can complement it, and you can do, this is a BioPython sequence. And you could also get the protein sequence. So if you translate from the start codon forward, and you can use the transcript objects to find out where that start codon is, so forth. All right, so that gives us the necessary information to figure out what a mutation means. So now let's start looking at some mutations. So I subsetted some of TCGA's ovarian cancer. So I think I took maybe 100 patients from their ovarian cancer set. And so you can load that MAF file, and you get back a variant collection, which is, am I really? I have 5 minutes. It's terrifying, it goes so quick. All right, well, so you get a variant collection, which is a container for variant objects. And it has a lot of different properties for grouping and filtering variants. And you could find out what are the most commonly mutated genes in ovarian cancer. And not surprisingly, TP53 comes back because it's sort of necessary to become a cancer. And you could also find out what are the protein effects of these mutations, meaning how did the mutations affect the proteins that get translated from specific transcripts. There's a whole vocabulary of these things. Is it a substitution of one amino acid or another, or is it a frame shift that might generate a whole novel sequence of amino acids? And so in this example, we look at the worst effect among those TP53 mutations, meaning the one that changed the protein the most, and it's a frame shift, and you could see that it took the whole tail of TP53 and replaced it with an entirely novel sequence. That's my presentation. These are open source projects. We like contributors. We like people to use our software, so please go ahead. And if there's any questions, I'll take them now. So what's very common in bioinformatics pipelines is a lot of this functionality is sort of patched together in some shell scripts. So there's not really a Python library that does the same thing, as far as I know. I might be wrong about that. I'll get called out if I'm wrong. But what I've seen, at least inside of Mount Sinai and some other places, is that bits and pieces of this functionality are strung together. You have a shell script that downloads some GTF files and some FASTA files, and then an R script that does something, and a Perl script that does something else. So the idea here is to give everything a more unified interface and to bring it together within a single Python library, because I prefer to do this kind of work when I'm exploring some functionality to do it all for my Python. I don't want to have to use multiple languages.
And this is it human only, or?
So it's currently human only. It's become a huge limitation because the cancer vaccine that I mentioned, they're doing preliminary work in mice. So we have to make predictions for mice, which means we have to go back through this whole stack of tools and generalize it to work across different reference genomes. So that's something that we're currently working on, is to lift that sort of human restriction. So that'll happen in the next month. Right now it's human only.
Lift it to just cover human and mice?
To be able to take arbitrary metadata files so that if you had a reference genome and an annotation set for some other species, you could use that too. Are there other questions?
Take another question while the next speaker is setting up.
Are there issues with size of BCF file? For example, have you used this software with 1,000 genomes?
So yeah, there are huge issues with size. We don't have a great story for that right now. So what happens is, we use a lot of other libraries like PyVCF to load the data, and it just takes a really long time. We've had a couple recent fixes to speed up just loading, but when it comes to the prediction of protein effects, that's still deadly slow. So I think we can load 1,000 genomes now, but if you wanted to look at all the protein effects, it would take you a couple days to get a result back.
I'm not Jimmy.
Okay, yes.